here we go. This unit was about cellular energetics, in which case we covered both cellular respiration and photosynthesis. A key part of understanding both of these processes is understanding ATP. ATP stands for adenosine triphosphate. It is this molecule right here. You will store energy between the second and the third phosphate of ATP. So we'll essentially break that bond in order to generate energy. That is what we do. In order to turn ADP, which is adenosine diphosphate, or two phosphates, into this energy-carrying molecule, we need an enzyme called ATP synthase. So in our bodies, we are constantly cycling between ADP, or non-energy storing adenosine diphosphate, and ATP, energy storing adenosine triphosphate. Cellular respiration is the process of trying to make ATP. That's the point of cellular respiration. What will happen is we'll take glucose, C6H12O6, we'll breathe in 6 oxygen molecules, and we'll release 6 carbon dioxide molecules, release water as a byproduct, and make ATP, which is the point of it. The first step of cellular respiration is glycolysis. Glycolysis is an anaerobic process, meaning that it is going to take place in the cytoplasm, but you don't need oxygen in order to do it. In glycolysis, we're going to split glucose, which is what glycolysis means. So we'll take our glucose molecule and make 2 pyruvate molecules. As we do this, we'll convert NAD plus into NADH, and we'll turn ADP into ATP twice for a net gain of 2 ATP. Normally, throughout cellular respiration, you will then take the pyruvate and bring it into the Krebs cycle, but I want to talk about fermentation real quick because it's really important. Fermentation is essentially just glycolysis. Lactic acid fermentation is a process of turning glucose into 2 pyruvate, the exact same as glycolysis, making 2 ATP, turning NAD plus into NADH. Where it differentiates itself is we are going to now turn pyruvate into lactic acid, which is going to allow us to turn NADH back into NAD plus. So we're making lactic acid as a byproduct. We don't want it. It sucks. It's poison. It causes fatigue and muscle soreness. But now we're allowed to use this NAD plus to go find another glucose and refire this process again, giving us 2 more ATP. The same thing applies in alcoholic fermentation. We do our glucose, split it into 2 pyruvate, make 2 ATP, make NADH, but now we'll turn pyruvate into both ethanol and carbon dioxide, which allows us to once more make our NAD plus and go find another glucose to redo this process. Fermentation is incredibly inefficient. You're only making 2 ATP per glucose molecule. So that's why we normally do cellular respiration. It just requires oxygen. So let's go through it. So after we make our pyruvate, in the presence of oxygen, we'll enter the mitochondria via the Krebs cycle. Acetyl coenzyme A is going to ward us through this process, in which case we'll turn NAD plus into NADH, FAD plus into FADH2, turn more ADP into ATP, making 2 more ATP, and then releasing carbon dioxide as a byproduct. We can then take our 2 high energy electron carriers, our NADH and FADH2, and bring them to our electron transport chain. This is going to take place in the mitochondrial membrane, and essentially what we're going to do is we're going to turn NADH into NAD+, FADH into FAD, which the purpose of this is that we're going to move our hydrogen ions above the mitochondrial membrane, and we're going to move electrons this way. Once we have created this heavy concentration gradient, these will flow through ATP synthase, which is essentially going to spin and start doing this process right here, turning ADP into ATP. This will happen 32 times. So overall, we have made 36 ATP, 2 from glycolysis, 2 from the Krebs cycle, and 32 from the electron transport chain, showing how much more efficient aerobic cellular respiration is than anaerobic cellular respiration. Let's move on to photosynthesis. In which case, photosynthesis is literally the opposite of cellular respiration. Our goal is no longer energy, our goal is food in terms of glucose. So it's going to bring in carbon dioxide through the stomata, bring in water through its roots, and bring in sunlight through absorbing that sunlight, and it's going to produce glucose and release oxygen. Oxygen is the byproduct. The way it's going to bring in the sunlight is it's going to absorb certain waves of visible light, and that's going to depend on what accessory pigment is part of it. Chlorophyll is going to reflect green and absorb red and blue light. Xanthophylls are going to absorb more green light and reflect yellow and brown light, and carotenoids are going to reflect orange and red light, absorbing more green light. No matter what, this is going to take place in the chloroplast. There are many different parts of the chloroplast that are important. The main ones I need you to know are the thylakoid membranes and stacks of thylakoid called granum, and then also the stroma, which is the intracellular space within a chloroplast. The light reaction is going to be the first part of photosynthesis. It's going to take place in the thylakoid membrane. So every single thylakoid stack of granum within one membrane, what we're going to do is we're going to absorb sunlight into photosystem 1 and 2. Both of these are going to be a large amount of chlorophyll, and they're going to be a protein channel, in which case we'll turn water into oxygen, releasing our oxygen. And we're also going to create this huge concentration gradient inside of the thylakoid membrane, turn NADP into NADPH, and then turn ADP into ATP by allowing hydrogen ions to flow to ATP synthase the exact same way they did over here in the electron transport chain. So at this point, we now have ATP and NADPH, and we're going to move on to the Calvin cycle, or the light-independent reaction. This is going to take place in the stroma, or the intracellular space. So what's happening in the Calvin cycle is we're going to have 3 RUBP, or 5 carbon sugars. These are present in your cells already, not in your cells, in the chloroplast already. We're going to fixate carbon dioxide with an enzyme called rubisco. Rubisco is going to bring in those carbon dioxide molecules into the RUBP, and they are going to split into 2 PGAs each. Overall, the point I'm making is we'll have 6 PGA sugars, or 3 carbon sugars. That was our fixation process. Then we are simply going to take that PGA and reduce it into PGAL, or also known as G3P. The reason we're able to do that is because NADPH and ATP allow us to reduce that. Remember that we got the NADPH and ATP from over here. So then now we have 6 PGAL, or 3 carbon sugars. We need 2 of those to make our glucose end product. So we're going to take 1 out right now, because that's all we can afford, and we're going to turn 5 of them back into RUBP. So we're going to use ATP and regenerate them back into RUBP. So we're back at this step right here. What we need to do now is turn the entire thing one more time. So bring in another 3 carbon dioxide, reduce the PGA, reduce the PGAL, and then we'll release a second PGAL to make a singular glucose molecule. That is the process of how we make glucose in the Calvin cycle. And that is all you really need to know for the test. That's it.